everybody hear me? Okay. So, yes, my name is Yuxing, and my topic today is actually talking about discovery of natural antimicrobial from the wooden cheese board. So, yeah, I would take it off. Yes. Okay, that sounds better. As we know, our lab is actually studying on Listeria monocytogenes. So, Listeria monocytogenes is a prominent foodborne pathogens. So as we know, it's a causes of listeriosis. And right now, many listeriosis outbreaks that has been associated with dairy products. And then people start to wondering whether listeria can actually survive in the cheese ripening process is because it has been reported that listeria is highly resilient and persistent. And if it has a chance to contaminate in the food processing process, it's very hard to get rid of. And then the people start wondering, is there any possibility that Listeria can survive in the cheese ripening process? Is due to a tour in the cheese ripening process that widely used in the cheese making factory. It's wooden board. So wooden board is very essential for cheese ripening process due to the porosity of the wood and the natural characteristics. So most important reason for wooden board are essential for cheese ripening process is due to three characteristics. First, the porosity of the wood will make a perfect moisture reservoirs for the cheese, which means if the cheese have excess moisture, and the wood will hold the moisture, and it will return back to the cheese when the low moisture prevails. And also because of porosity of the wood, it can provide a place for a specific microbe to establish themselves and receding onto the surface of the cheese during the cheese ripening process. And what's more important reason is that many cheese makers believe that the wooden boards can increase the flavor of their final products. So that's why the wooden board are widely used in the cheese ripening process. So whether or not the pathogens can grow on those wooden board surface or not, due to the porosity of the wood, and it's the hardness to and it's very hard to clean the wooden board as well. So to following up this idea, our lab did want to study Listeria behavior onto the cheese board surface following inoculation. So we request some cheese board from the cheesemaker, different cheesemaker in Wisconsin. And then all the cheese board that we receive, we can distinguish them to two sections. One, we call it cheesy sections. It's a section we can see the cheese will place on. And other places of the cheese board, we call it woody section. So we inoculate Listeria onto top of two sections, and we study Listeria behavior during 20 days. And here is one of the representative data. As you can see here, the top two black curve represent the total box we recover from cheesy and woody section, and the bottom two red curves represent Listeria population recovered from cheesy and woody section. The top two curve will tell us whether our recovery method is appropriate. And as you can see, the total box recovered from cheesy or woody section is pretty stable during the 20 days, means our method of recovery is appropriate. And also during the 20 days, there's enough nutrients or the environment is suitable for all the bugs to survive onto the cheese board surface. But if you look at Listeria population recover from cheesy and woody section, in the first 8 days, Listeria population will decrease about 2 logs and keep quite stable during the leftover 12 days. So it declines on the surface for the following inoculation, and it's inhibits onto the surface in the leftover days, according to this figure showing us. And this is only one cheese board, so we're thinking maybe we can test different type of cheese board used for different type of cheese fermentation. And then this is a whole data set. No, actually, this is part of the cheese board that we did. So there, we test the cheese board for use for softer cheese and use for hard cheese, and also the cheese board after cheese ripening process, it will wash. And then we test the washed cheese board as well. As you can see from all those three figures, it shows the same trend for Listeria population. So in the first 8 days, Listeria decreased about 2 logs and then was totally inhibited onto cheese board surface. And then that made us wondering what exactly the reason that inhibit Listeria. And then as we can imagine that there are two factors that actually participate into the whole process. So first is wood itself. So as we know, it has been reported that wood has a lot of antibacterial factors that could be the reason that inhibit Listeria. Also, since we inoculate Listeria into a community, since the cheese spores still have a microbiota survive onto the cheese spore surface, and if Listeria inoculate in that community, there could be potentially microbe that interact with Listeria by inhibiting Listeria to survive in this community. So to following up the idea, first we want to study whether it's the wood itself that inhibits Listeria by some antibacterial factors. To study on that, we repeat the same experiments, but instead of using the cheese board that using the cheese ripening process, we purchase some natural wooden board. These are a type of wood that widely used in the cheese making factory, cedar, pine, and spruce. So those are just a natural wooden board. They didn't get into the cheese ripening process at all. And then we did the same thing. We inoculate Listeria onto the surface, and then we assess the Listeria population at half an hour and after 4 days. As you can see, different type of wood will have different behaviors. But then after 4 days, Listeria either is not detected or is significant decreased after 4 days. So the wood actually has inhibition for Listeria. In the next step, we're trying to extract something from the wood itself and to see what exactly the antibacterial factor is. And then what's more important for us is that we are actually interested in the whole community, which is the cheese spore microbiota. Because how Listeria establishing a community and then survive onto a cheese spore surface is a question we want to ask. So before that, we actually did 16S region sequencing for all the cheese spore that we received. And then we will try to see whether they have different microbiota composition for all the type of cheese spore. So this is top 10 taxa among all the cheese spore that we receive from cheese makers. As you can see, the top 10 taxa all belongs to 3 main phylum. One is actinobacteria, firmicutes, and proteobacteria. But what's more interesting is that from different cheese makers, they have different abundance. And even within the same cheese maker, washed cheese board and unwashed cheese board also have different abundance for different genuses. So they do have a diverse microbiota. And then that gives us an idea, maybe we can isolate some microbes, maybe have a potential inhibition for Listeria. To following that idea, we isolate based on the colony morphologies, and then we test the inhibition for Listeria by spot those individual microbe onto Listeria lung, and then if they can create a zone of clearance, we go forward and identify the species. So there's 3 ways that we actually try to identify the species that have inhibition for Listeria. The first screening is just by multi-top. It will give me the basic species information, and then we did 16S RNA sequencing, and in the end, I choose 2 strains that we think is interesting, and we will send out for whole genome sequencing and focusing more on those 2 strains. One is Bacillus subfensis, another is Serratia macescens. So for Bacillus subfensis, what exactly the inhibition for Listeria look like? We find out that the Bacillus subfensis can secrete the anti-Listeria factors into a cell-free supernatant. As you can see from here, this is Listeria 10403S treated with different percentage of Bacillus cell-free supernatant. As you can see from this figure, if you treat the Listeria with higher concentration of Bacillus supernatant, Listeria was inhibited in that condition, especially if you treat it with 50%, there's no Listeria growth was detected. And then we also find out that the last figure was testing for Listeria. Listeria have different strains as well, and 10403S is a standard lab strains, and we also tested some other Listeria strains as well, such as LO20A and F2365. They're all clinical Listeria strains. As you can see, they all have the same trends if we treat it with Bacillus supernatant, even at different percentage, their relative growth rate was decreased. And then we also find out that this antimicrobial can also inhibit other gram-positive bacteria. Except for E. coli, which I point out here, it's the only gram-negative bacteria we tested in the lab, and then it does not have a significant inhibition during that condition. So following up that idea, we also find out that this actually the secreted antimicrobial also can kill Listeria. So the figure I just showed is actually the relative growth rate. So we only know that Listeria was not detected, but we don't know it's inhibited or actually killed. So to figure out whether it's killed Listeria, I have to assess the Listeria population from the whole system. As you can see from here, is this Listeria treated with Listeria its own cell-free supernatant, or Listeria treated with Bacillus supernatant. And then I assess the population of Listeria after 20 hours of treatment. As you can see, after 20 hours, if Listeria was treated with Bacillus supernatant, there's no Listeria was detected after 20 hours. And I also find out if I heat the Bacillus supernatant first and treat with Listeria, the killing effect was lost. So that make me feel like this probably potential antimicrobial is a protein or not. So to following that idea to test whether this antimicrobial is protein or not, so I treated with protein SK. As you can see, before the treatment with protein SK, the Bacillus subfuscis supernatant can create a zone of clearance on Listeria lawn on the left panel picture. And then if I treated the supernatant with protein SK and I spot the same supernatant, like protein SK treated supernatant, the zones of clearance disappear. So these derived antimicrobial factors are proteins. And then since we know it's proteins and we want to identify what exactly the anti-Listeria protein it is, to figure it out is the easiest way or is common way is to generate a mutant that does not have inhibition. So to compare this mutant with wild type, especially maybe we can do a proteomics to compare the secretome of the mutant and the wild type to figure out what exactly their antimicrobial protein it is. So to following up this idea, we actually did proteomics for those mutant and wild type. So as you can see from this figure, the y-axis represent the log2/4 changes of those different proteins abundance from the mutant versus wild type. And then we are looking for the one that is significant less abundance, not significant, sorry, it's less abundance in the mutant compared with wild type. So we're looking for the -4 change. As you can see, there are several candidates pumps up in my proteomics data. Most of them belongs to lipase and peptidase and proteinase. And also what's more interesting that there's some type 7 secretion system effectors also shows up in my proteomics data. And then in the next step, I'm trying to express those protein into E. coli host and see whether they actually is the anti-Listeria proteins or not. And then talk about Bacillus subfuscis, and as we know, our lab's still interested in Listeria. So to study Listeria, we interested in how Listeria responds to those antimicrobial proteins. What exactly Listeria will do if a have this attack from Bacillus. So to study on that, we did RNA sequencing for Listeria. After Listeria treated with Bacillus supernatant for some time, and then we study the RNA sequencing and see Listeria transcriptome changes. As you can see, this is RNA-seq data from my last experiment, and then for the upregulated genes, there's something interesting showed up, which is called glutamate decarboxylase. So this glutamate decarboxylase belongs to a gas system. So the gas system is very important for mediating the pH homeostasis in Listeria. So, so the gas system is important to decrease intracellular hydrogen ion concentration in Listeria generally. So we are thinking maybe this anti-Listeria factors actually changing the pH environment so to inhibit Listeria. But then we haven't kind of verified this right now. So in the next step, I'm trying to verify this hypothesis. And then talking about serratia mastitis, as I talked about, we also did whole genome sequencing for those strains as well. So what exactly serratia mastitis inhibit Listeria? So different from Bacillus subfuscis, serratia mastitis can outcompete Listeria into the co-culture system. So as we mentioned about co-culture is that we co-culture those two strains in the same system or inoculate them alone to see whether they're inhibited in the co-culture system. As you can see here, the left panel represents Listeria growth in in the co-culture system, and then versus Listeria growth in the monoculture. And the right panel is Serratia growth in the co-culture system versus Serratia in the monoculture. As you can, if you look at the 100% BHI broth, it says 20 hours Listeria log 10-fold change. It's about 2 log changes after 20 hours in the co-culture system. But Serratia does not have any growth defects in the co-culture system as well at all. And then the defects of co-culture system assay is that you cannot tell this inhibition is nutrient related or not. So we are trying to see whether this co-culture inhibition is nutrient related. So we try to use different strengths of BHI and see whether it's related to nutrients. As you can see from here, if we decrease the richness of BHI to 20%, the inhibition was enlarged in the co-culture. You can see the 25% Listeria is about almost 4 log reduction in the co-culture system compared with 100% is about 2 log reduction here, but Serratia does not have any defect in the co-culture, even you reduce the richness of the BHI broth. So the co-culture inhibition does relate to the nutrients, but it there means there's no other antibacterial functioning. We found out it actually has antimicrobial happening in the whole process. As you can see from this figure, this is just spot Listeria in the presence and without the presence of Serratia. As you can see, if with the presence of Serratia, Listeria cannot grow super close to Serratia at all. So it's actually Serratia can secrete antimicrobial to inhibit Listeria. And then I start to extract the antimicrobial from Serratia and then treat it with Listeria with different percentage of the antimicrobial extract. Even at lower, about 12.5% of extract, Listeria population cannot be detected after 20 hours. But Serratia mastitis is kind of widely studied environmental opportunistic pathogens. So there's several known antimicrobial that was found from Serratia mastitis. One of them called prodigiosa, another one is Serratomoli. And then we cooperate with another lab in Pittsburgh, and we request those mutant that from their lab. So prodigiosin, the deletion of prodigiosin or deletion of Serratomoli or the double mutant deletion. And we spot those strains onto Listeria lawn, and we find out that even the single deletion strains or the double deletion strain all can create a zones of clearance on Listeria lawn. So this actually, the thing that we saw, they inhibit Listeria is something that has been discovered yet. And then so in the next step, we're trying to figure out what exactly the active compound from Serratia. Also, we're going to do the same thing. We're going to generate a Serratia mutant that does not have inhibition for Listeria, and then we try to compare their differences with wild type. That's all for today, and thanks to my advisor and my lab mates for all the support, and I'm happy to take any questions. Thank you. Okay.
So when you were talking about the screen, can you walk me through what I needed to do?
You mean the Bacillus one or Serratia one? So actually, I generate a mutant first. So like by EMS mutagenesis. So EMS is changing point mutation from GC to AT, and then I screen for the mutant that does not have inhibition. And then I screen that mutant out, and I did proteomics. So it's a comparative secretome of the mutant and the wild type. Yeah, so actually get the mutant first, and then do the proteomics.
Did you ever consider using a transposon screen for that?
Yes. So the thing is, there are different ways of mutagenesis, but then I think the easiest way for Bacillus, all the literature is using EMS. It's just easier chemical ways, right? And then for transposon mutagenesis, we're going to do this in Serratia mastitis. It's just different bacteria, which way is easier to get the mutants out.
Thank you.
I'm sorry, I think we have to go on to our next speaker.
Thank you, Yuxing. I'm the co-moderator. I'm Joe Pierre. I'm assistant professor of nutritional sciences and surgery, and a member of the Dairy Innovation Hub. So our next speaker today is Rachel Dietrich from the Gerard Lab, and they study food protein chemistry. She obtained her bachelor's degree in chemistry and mathematics from Roberts Wesleyan College and her PhD in chemistry from UW-Madison. I believe today we're going to be hearing about demonstrating the value of dairy ingredients and protein and fiber enriched snacks. So please join me in welcoming Rachel.
Thank you for that introduction. So yes, I'm presenting my work today on demonstrating the value of dairy ingredients in extruded snack foods. So we know that demand is increasing for protein-rich and fiber-rich foods. So the majority of consumers when grocery shopping look for foods that are high in protein. And some consumers are also looking for foods with bioactive proteins that offer additional health benefits. Additionally, most consumers do not eat sufficient dietary fiber. So if we can add fiber to popular protein-rich foods, then we could improve public health. Whey co-products are an underutilized source of these nutrients. So whey is the byproduct of cheese production, and it's further processed to produce two co-products: whey protein phospholipid concentrate, or WPPC, and D-lactose permeate, or DLP. WPPC contains 50% protein, and 23% of these proteins are bioactive proteins from the milk fat globule membrane. DLP contains 60% lactose, which can be polymerized to form fiber, and I'll talk more about that later. To give you an idea about how abundant these are, in 2022, US dairy farms produced 14 billion pounds of cheese, which resulted in 20 billion pounds of WPPC and 70 billion pounds of DLP. Yet these abundant and nutritious co-products were primarily used in low-value products like animal feed and fertilizer. Therefore, we propose to increase the value of dairy whey ingredients by incorporating them into nutritious extruded snack foods. And so extrusion uses heat and shear to produce puff snacks. In our extruder, we put the dry ingredients in the feeder, and then they're mixed with water as they move along a screw. Our extruder has two parallel twin screws. They're heated, and then eventually they exit a small hole called a die. And as they exit the die, water evaporates, which creates that puff texture that you have in cheese puffs. This is a picture of the benchtop extruder that I'm using, and this shows what the extruded product looks like as it exits the extruder. Note that only dry ingredients can go here. We get the DLP in a liquid form and have to freeze dry it prior to extrusion. We also, in this process, want to polymerize lactose from DLP. And so before I get into my results, I'm going to talk about what fiber is and how we plan to make it. The US FDA defines fiber as carbohydrates that are not digestible. If the carbohydrates are synthetic, which would be the case for my work, then they have to have a demonstrated health benefit in order to be defined as fiber. Fiber includes resistant starch as well as non-digestible oligosaccharides. So those are two things that I'll measure for the puffs that I make. Previous work has shown that lactose can be polymerized to form fiber. It's also been shown that lactose typically isn't polymerized directly, but it's first hydrolyzed into its component sugars, and then those sugars undergo condensation reactions to create polymers. And so this is the process that we want to use to create fiber from DLP. And so for my puffs, I made two different recipes, one that has 20% DLP and one that has 40%. Both of these have 20% protein coming primarily from WPPC, and they should have between 7 and 14% fiber, assuming that I achieve similar levels of lactose polymerization that have been previously obtained. I tried making puffs with normal DI water as well as water adjusted to pH 3.5 to try to increase the polymerization. And after extrusion, I dry the puffs at 37 degrees. I also made puffs with two different feeder rates to see how that would affect the structure of the puffs. The feeder rate is just how quickly the dry ingredients are fed into the extruder. And so these are how I label the 4 types of puffs. 1 indicates that it has 20% DLP, 2 is the 40% DLP, and then the 25 or 30 are the 2 different feeder rates that I used. So here are pictures of the puffs that I made. You can see that for puffs with 20% DLP, adjusting the feeder rate increased the expansion for those puffs. But for 40% DLP, they showed similar expansion regardless of the feeder rate that I used. And so here are those results shown graphically. The y-axis is the expansion ratio. That's just the ratio of the puff diameter compared to the diameter of the die. And what we see is that overall, all the puffs showed similar levels of expansion, regardless of if they had 20% or 40% DLP. So how do my puffs compare to commercial puffs? For this, I compared my puffs to 6 different types of commercial puffs, 3 corn-based puffs, Cheetos, Kroger, Puff Snacks, and Cheetos Crunchy, and then 3 different types of puffs with added protein. I look at both physical characteristics as well as nutrition content. And so for size, I measured the diameter, and I found that for commercial puffs, there's a range in how big those puffs are. So for example, Cheetos are much larger than the Cheetos Crunchy. The puffs I made are on the smaller end of what's sold as commercial puffs, but they're pretty similar to like a Cheetos Crunchy snack. Next, I looked at texture. So for this, I use a texture analyzer where a flat plate compresses the puff and measures the force generated upon compression. And so I'm defining hardness as the maximum force experienced during that compression. And so here's what I found. When I increased the DLP content, that significantly increased the hardness of the puffs. Additionally, all of my puffs are harder than the corn-based commercial puffs. However, the puffs with 20% DLP are similar hardness compared to commercial protein puffs. So this does fall in what would be considered a typical texture for this type of product. So what about nutrition content? My puffs have 6 grams of protein for a 30-gram protein size. This is similar to some protein puffs, like these Iwan organic protein puffs, and they can be labeled as a good source of protein because they contribute 10% of the recommended value. Additionally, my puffs may offer additional health benefits because the protein content comes from WPPC, which is rich in bioactive proteins. And so I wanted to measure, do I still maintain bioactive protein after extrusion? And to measure this, I measured xanthine oxidase activity. Xanthine oxidase is an enzyme that converts hypoxanthine to uric acid. And for me, I used it as a marker to measure what the overall protein activity may be. I find that xanthine oxidase activity is 8.5% after extrusion of the starting activity in WPPC. And so I am maintaining some of that protein bioactivity after extrusion. Now, if we wanted to create a higher value puff product with additional bioactive protein content, we could increase the content of WPPC, or we could add in additional components that include bioactive protein content. And so then what about fiber? Most commercial puffs do not contain any fiber. Out of the 6 I looked at, only Iwan organic puffs contained 3 grams of fiber, which does make them a good source of fiber. And so what about my puffs? To measure the fiber content, I first measured resistant starch. And to do this, I subject the puffs to digestion with 2 enzymes, and then I pull out aliquots at different time points and measure how much glucose has been formed from the digestion of starch. Any starch that's not digested after 4 hours is considered resistant starch. I found that extrusion does not generate any resistant starch. So here, this shows the starting starch composition. With some resistant starch, the bulk is slowly digestible starch that takes between 20 minutes and 4 hours to be digested, and then some rapidly digested starch, which is digested before 20 minutes. This starting resistant starch comes from cornstarch. After extrusion, almost all of the starch is converted to rapidly digestible starch. And this is true both for puffs with 20% DLP and puffs with 40% DLP. And it's true whether I extruded with normal DI water or water adjusted to pH 3.5. And so next I wondered, well, am I producing any oligosaccharides? And so to look at this, I used HPLC and an RI detector and a column that can separate lactose, glucose, and galactose. Oligosaccharides will show up at lower retention times. So here I'm showing oligosaccharides I generated from a mixture of glucose and galactose heated in the presence of citric acid. And I can hydrolyze these oligosaccharides to get back the component sugars. And so with my puffs, I do see some oligosaccharide content from this peak at that lower retention time. And if I hydrolyze this sample, these are hydrolyzed to produce back glucose and galactose. This glucose peak is much higher because I am adding in glucose into my puffs, and also the cornstarch is hydrolyzed to produce glucose. Additionally, I have this peak here that could be non-hydrolyzed oligosaccharides, or it could be another degradation product that's produced from the hydrolysis. So I'm currently working to figure out what that is. Similarly, with the 40% puffs, we have oligosaccharide peaks that are hydrolyzed to produce glucose and galactose. And additionally, we have this peak here that I'm working to identify. And so in summary, we demonstrate the feasibility of using whey co-products to produce nutritious extruded snacks. The DLP and WPPC mixtures do produce extruded puffs that contain 20% protein, and they have a similar size and hardness compared to commercial protein puffs. Extrusion produced some oligosaccharides, but not yet resistant starch. And so moving forward, I want to increase the yield of non-digestible oligosaccharides, which I plan to do by hydrolyzing lactose in DLP and polymerizing it prior to extrusion, and then taking that polymerized product and using it in extrusion. I then want to be able to identify whether any of the oligosaccharides I'm making have been shown to produce health benefits, because that will be a necessary step in order to label it as dietary fiber. And so achieving these steps will enable me to make puffs that are a good source of both fiber and protein. And so with that, I want to acknowledge all the people who have helped me with this work. I want to thank my advisor, Audrey Gerard, for her guidance on the project, other members of my lab, Lily Lincoln, who trained me on the extruder, and the undergraduate and high school students who helped me to collect this data. Thank you to our collaborator, Gulistan Öztürk, for sharing her expertise on bioactive proteins in WPPC. Thank you to Jackie Cooper in the environmental engineering facility for training me on the HPLC that I used. And then our industry partners, they have shared feedback on our work multiple times throughout the project, and also provided the DLP and WBPC I used for this work. And finally, thank you to the Dairy Innovation Hub for providing the funding for this project. So thank you for listening to my talk. I'll be happy to answer any questions, and also you can find me at poster number 29. Yeah, I'm using it as a lactose source, but also I want to use DLP because it's currently only used in low value products. And so using it for a higher value product like a snack food would increase its value, and it's a widely abundant ingredient that is just available, and we're just not using it very much now. Yeah, so it's like 60% lactose, and then it has minerals and yeah, other things in there. I did measure moisture content. Yeah, that's a great question. My moisture content, I didn't show the results here. It was higher. It might have been like 10%. It was higher than what commercial puffs are. But I think I can improve that either by adjusting the water rate during extrusion or just drying for a longer time. Likely, yeah, it would. So yeah, DLP is very salty, and you can definitely taste that. I think adding other flavor components like a cheese flavor would definitely help that. But that's definitely something to think about, as I'm not sure the 40% DLP are gonna really be good to eat. Yeah.
Pleased to announce our third speaker, David Lang. He's a graduate student here at UW-Madison pursuing a master's degree in food science under Associate Professor Brad Holig. He got his BS degree here as well in food science as a graduate student. He's working towards the goal of streamlining the analysis of the small molecules in dairy products via liquid chromatography mass spec. So please welcome.
Well, great. Yeah, my name is David Lang. I'm a graduate research assistant in Dr. Bowling's research group here on campus. And today I'm actually talking about a subject that I actually successfully defended my master's thesis on a couple of weeks ago, but liquid chromatography, tandem high resolution mass spectrometry as an analytical method for dairy metabolomics. And I know that's kind of a mouthful, and I'm really thankful for the keynote presentation this morning because I think it actually ties in pretty well where what was discussed this morning. This project kind of elucidates, how we detect those sorts of things. So since it's kind of a mouthful, and I always like to start with what does metabolomics actually mean? So metabolomics, defined in this formal definition, is a systematic study of the unique chemical fingerprints that specific cellular processes leave behind. This is essentially an analytical approach to determine, qualify, and sometimes quantitate all of the chemical compounds within a given sample. The thought process behind doing this is to kind of provide this greater understanding of the underlying cellular processes in cellular systems. But this can actually kind of extend further into how is processing impacting the chemical makeup of samples, and how might cultures be influencing what's present, that sort of thing. The limits to what these types of analytical methods and analytical tools can provide is really quite enormous. While that is the formal definition of metabolomics, the actual definition of metabolomics is a little bit fishier. A lot of people actually like to liken it to kind of this like a deep sea fishing, where ultimately you're not going to be able to catch every fish in the sea using the same approach. Unfortunately, right now, at this point in time, there's no kind of catch-all method for determining all of the compounds in any given sample. So ultimately, the approach you use is going to kind of determine what you're going to find. Whether you're going to search specifically for certain fish in one area, or you want to cast a large net and pull up everything. But when you cast that large net, maybe there's going to be really high abundances of certain things that you have to kind of sift through and kind of see what's there. But ultimately, through kind of these processes, it can be really useful for kind of determining, what is always, naturally like high in abundance in these samples, and what may be influenced based on the environment, based on the processing factors. How does this really tie into dairy, though? Well, in dairy, if you really think about it, dairy products are a very complex chemical equation. They have very major impacts from the primary inputs into the dairy cows will have a very large impact on its first primary output, so the chemical composition of the milk samples. Honestly, you could probably even put something right here based off of how you heat treat the milk as well. So whether you're using HTST, UHT, ultrafiltration, or you're just looking at the raw milk, all of these primary outputs from the cows will have very different chemical compositions, that whole chemical makeup. But what's even more interesting to realize is that the makeup of this primary output from the cows is going to have very significant impacts on a lot of the downstream processes and downstream products of milk. So the thought process in this project is really to kind of create these analytical methods to determine, what chemically speaking constitutes a cheese from a cheese, a yogurt from a yogurt. What chemically speaking on a small molecule basis is what differentiates these products from one another. We're starting to see it a bit more in literature. It's becoming a lot more ubiquitous, but a lot of the times in these metabolomic studies, you run these methods using various different types of analytical tools, whether it's NMR, LCMS, GCMS, any of that. You get so much data that you have to usually pair it to a large number of multivariate analyses. But once you kind of start deconvoluting that data, these studies can actually really help signify, markers of milk quality, predictions of authenticity, evaluations of technical properties, and kind of discrimination of feeding systems. So these analytical tools have actually been proficient in determining, the origin of where cheeses are made in Italy based on their region, or determining, whether or not a milk is from a grass-fed cow or a conventionally fed cow, just based on these chemical signatures. For example, here's another paper over here, which was able to distinguish UHT milks from pasteurized milks and raw milks. And what's kind of underlying these data points in this principal component analysis is that whole metabolome, so that whole chemical makeup of each of these samples. They're using these large sets of data essentially to authenticate samples based on process in this case. So again, the project objectives here were developing methods of untargeted metabolomic analysis of dairy products, encompassing all these different categories, ultimately for the goal of analyzing them and banking this metabolomic data. There's no shortage of analytical techniques for analysis of all this data, especially when it comes down to like differential analysis, determining what's unique and what's different. And you could spend a long time doing that, but I'll just kind of touch over a few of them in the aspect of time in this presentation. But I think most importantly from this project as well is banking a lot of this data. So in metabolomics, it's kind of a hairy subject right now, and it usually relies on the presence of already analyzed samples to validate what you're detecting through these processes. So a major research output from this project is going to be the publication of the databases from the study for public use. So in comparative analyses, once you get to this is skipping a large step of like how you perform the extraction, how you perform the separation, and how you perform the analysis. And I'm happy to speak about that later on. But I'm getting right into the meat of it, kind of what did we really pull out of this, and what you can do with the data. So again, using PCA, so principal component analysis, to kind of authenticate samples and determine, what are some of those differentiation factors. Hierarchical cluster analysis, that's what I'm primarily going to be speaking about in just a moment here. This provides us an indication of how closely metabolomically related different dairy samples are to one another. And then if you were interested in looking directly at the relative abundance of an individual compound, a trend chart feature would be a good application for that. So what does this actually really look like? So getting to the hierarchical cluster analysis, that's what provides us these heat maps here. To kind of understand what's going on here, each of these individual bands is a individual detected chemical compound. And the color designates, what that relative abundance is compared to all the other samples. And this is actually two parts of one giant figure right here. So if you look at this on a really large scale of comparing tons of different dairy products to each other, it's a little bit messier because obviously, as we were talking about, as we learned about this morning, dairy matrices can be very distinct from one another. Milks are obviously very different in their makeup from cheeses. So you get to see some pretty large differentiations between these samples. But despite that, you can still see quite a bit of distinctions based on our key groupings of these samples based on their whole metabolomic fingerprint. You'll notice that all of these darker blue ones, besides this one whey protein concentrate for some reason, are all categories of fluid milks, and they are all metabolomically distinct to each other than what we have down here, which are powdered, or dairy powders. So whey protein isolates, whey protein concentrates, hydrolyzed whey. You'll notice in the hydrolyzed whey signature, this really dark red band of compounds that are really high in relative abundance compared to everything else. It's not surprising that this dark red band is mostly free amino acids, which is what you'd exactly expect from a hydrolyzed whey sample. When we go down to the other side of this figure, you can start seeing, how closely related are these types of cheese samples to one another on a whole metabolome basis. You start to see that based off of the linkages in this dendrogram over here, that the mild cheddar is most closely related to the 1-year age cheddar on a whole chemical basis before it's related to the Gouda, and et cetera. And you'll see, consistent metabolomic fingerprints that constitute all these cheeses. And right below that, distinct fingerprints that constitute the whole chemical compositions of yogurts, followed by yogurt powders, and so on with those samples. It's, as I said, it's a little messy looking at these on like this whole like dairy product comparison. So it's actually pretty useful once you start zooming in on these things. So if you zoom in to just these categories of things and you just compare within categories, you can draw some further distinctions on what makes these types of samples unique. Here we have these whole metabolomic kind of fingerprints for all these different types of yogurts. And not surprisingly, these strained yogurts are most closely related to each other before they're related to a lot of these conventionally prepared yogurts. You can take the time to zoom in and try to deconvolute all of these compounds, but I think ultimately it goes back to that fishing analogy where it's like, what are you, well, you need to first understand what you're looking for. So to kind of deconvolute that even further a little bit, a useful technique is performing targeted metabolomics and looking just at certain compounds specifically. And these platforms are really useful for that as well. So if you wanted to just look at the relative abundance of a set of compounds, you can do that. And you can perform that very easily, and you can draw some pretty incredible insights from that. So say, for example, you're interested in the relative abundance of leucic acid in yogurts, also known as hydroxyisocaproic acid, which is a suspected bioactive responsible for some of the health-promoting benefits of yogurt. According to some research, you would see that it's actually higher in relative abundance in, let's see, which one is it? Right there, higher in relative abundance in the strained yogurts, the skyr and the Greek yogurt, compared to the rest of these. Again, the applications for this just go on and on. It really depends on what you want to look for, but the intent of this project was just to develop these methods in the first place. Just for curiosity's sake, for anyone interested in cheeses, here again you can see kind of these whole metabolome fingerprints for all the different cheeses to understand how closely related one cheese is to another based on its chemical composition. And you'll see quite a bit of a impact of aging. Each one is kind of related to the next one. You get aged cheddar, aged Gouda, further aged cheddar is closely related to each other. And then distinct signatures for brie, a mold ripened cheese, or baby Swiss, which may use very different starter cultures than the cheddars or the mozzarella. Zooming in again, you can draw some distinctions from these samples. So for example, if you wanted to just look at, you had this targeted list of metabolites you're interested in, you would notice that in the brie, there's a really high relative abundance of free fatty acids compared to the rest of the samples. And in the baby Swiss, really high relative abundances of free amino acids. And then in some of the aged cheeses, you might find some specific metabolites, which may just be indicative of aging as a process. So beyond that, again, going into database generation, what's really great about these platforms is it's incredibly easy to generate databases and kind of publish them in a way that's going to be useful for a lot of downstream research. It's kind of an iterative process in these. So it's like once you build one database, you can reincorporate that into your next study, and it just kind of builds on itself. But essentially, you can build databases, whether it's based on individual samples within a category. So you wanted to build a Parmesan database of just Parmesan cheeses, you can analyze all of those, publish that. Or if you wanted to build a database that corresponds to the metabolites in all cheeses, you can do that as well. And these are all things that will be published later this summer. So just quickly in conclusion, yeah, with this project, I was able to determine that dairy products were metabolomically distinguishable. And these distinctions are going to serve, hopefully, as a foundation for a lot of future research applications. And these databases will be available in the future. In the next steps, again, just cleaning up a lot of these databases. There's going to be continued dairy product metabolite discovery. As I said, it's becoming really ubiquitous in industry. It's really cool to be on the kind of forefront of this and see it in action. And then future predicted metabolomic studies. So I'm kind of at time here, so I just want to acknowledge my lab group, Dr. Brad Bowling and Chong Wu, for their support, my colleagues in the Bowling Research Group, and of course, my wife Grace, and that's our dog, Pumpkin. So I'll take any questions now. It's a lot to say, so. Yeah, so this project is really kind of cursory. What will be really useful in the future is if you're interested in free fatty acids, optimizing a method just to target those and running standard curves associated with the ones that you're interested in quantifying. None of this is absolute quantified. This is all relative quantification at this point in time, yeah. Yeah, it can generally provide impact on how it's grouped, but for the most part, when you are looking cross-categorically, it's still going to group them that way. Yeah, ultimately it comes down, if you want to use like a Pearson or I'm trying to remember some of the other ones, but different ways to perform your linkage functions in dendrograms, absolutely.
Let's thank David one more time.
Okay, is this on? Gonna call everyone back. All right. So starting with our second hour, we're gonna move product and application innovations in the next three talks. So our first speaker here is Sonali Mohapatra. She's a postdoc in biological systems engineering at UW-Madison, mentored by Shejun Pan. She received her PhD in fermentation technology from BPUT University in India. And I understand we're missing a couple words that she's going to point out here to clarify the title. So she'll take care of that, but please join me in welcoming Sonali to the podium.
Thank you. So the topic which I'm going to speak about is chemically synthesized galactooligosaccharides from lactose in combination with milk protein, which enhances its prebiotic efficiency in selected probiotics. So we are just missing the from lactose in combination. So, okay. And I'm Sonali, and my co-authors here have also helped me immensely in this project, so I'm thankful to them as well. So starting with okay. So basically my topic, the broad topic that I'm going to talk about is prebiotics and probiotics. But before going to that, let's have a very quick look on what basically intimidated me to take this topic on. Now, if you can see, if we see the global probiotics market, it's around, which is this is a prediction which I've taken from the Market Insights 2024 report. So you can see that the 2023 probiotics market was around 70.95 billion USD and is predicted to go to 115.07 USD billion dollars, which is a CAGR, that is the compound rate of increase of 8.4%. That's very good. However, if we see the prebiotics market, you can see that it is just in a very naive stage at just starting at 6.95 billion USD and predicted to reach by 13.26 billion USD. But if you put a look on the CAGR, it is around 11.4%. So which is around 35%, approximately 35% more than the probiotics market. Now, what has basically led the market to go from the probiotic to the prebiotics? So the basic constraints of a probiotics are basically development of a probiotic strain, like starting from development of a probiotic strain from next-gen sequencing, going to see that how it, because most of the probiotic strains are antibiotic resistant, so that is also a concern that how that group is going to affect a particular batch of human beings. Third, it is very difficult to monitor, the persistence of this probiotics when it is reaching the general human beings or when it is given in a formula for a particular intake. Further, it also has that the probiotic administration ways, that is how it is administered into the person, also makes it very difficult because sometimes it may be contaminated. Further, it is also seen that the suitability of the probiotics in a vulnerable group and also in a healthy group also depends it's very variable. So seeing to all these constraints, the prebiotic market is basically where we can that is what is the reason why the market is mostly revolving to go towards the prebiotic. Now, advantages of prebiotic is that it can be found and synthesized from natural food and food waste. It have great shelf life with minimal chances, doesn't get altered while passing through the digestive tract, selective proliferation of gut microbes, that is, we don't need all the gut microbes to get enhanced with our prebiotics, so there's a very selective proliferation of that. Easy packaging and storage as compared to the probiotics. Further, with these things, the industrial drivers, you can see some examples I have given who are the Kellogg's. These are the renowned industrial drivers of prebiotics, that the Kellogg's, Cargill and Eagle Genomics, Abbott, and these all are, just to name a few, are some of the most important drivers of the prebiotics. So coming to the introduction, so I have 3 P's for you to introduce. The first P being the prebiotic, So the prebiotic, as I have already said, a little bit of introduction, but giving it a very scientific introduction is that the prebiotics are basically substances that do not get digested in our stomach and get into our small intestine, where they are basically utilized by our second P, that is called as the probiotics, for their sustainability. Now, while they grow with the help of this with the prebiotics, they also produce some short chain fatty acids, which is basically some compounds or the byproducts of their growth, which basically also helps in the enhancement of human intestine and also the immunity functions of the human beings. So what's the third P? So the third P is the protein. Now, why are we talking about protein suddenly here? Now, what happens is when the prebiotic is given, when a prebiotic formulation is made, it is never given as a solitary entity. It is always given as a mixture with, most of the time, the proteins. So for an example, the infant formulas are mostly packed with proteins, and that is where we use these probiotics to give that as a embedded structure. So the next question is, can the delivery of our prebiotic, that is the GOSH, that is galacto-oligosaccharide, with different proteins affect the growth of selective probiotics? Now, why am I talking about this? Because when the proteins are combined with this prebiotics, what happens is it goes through this system where we have the amylase in our saliva, the pectin in our digestive system, stomach, and also when the protein goes through it, so it basically turns into peptides. So that is where, the structure of all the proteins change, and the GOSH is also altered because that the glycan protein complex is changed. So taking these into consideration, so our work is basically focused on producing galacto-oligosaccharides from lactose. So lactose are basically the byproducts. We can obtain lactose from the whey permeate, as some of the speakers have already mentioned. So we produce galacto-oligosaccharides from lactose by a hydrolysis followed by glycosylation in a concentrated acid. Followed by, we do some in vitro experiments to test the application of our produced galacto-oligosaccharide. In order to verify that how our in vitro experiments are particularly evident, that is that how our GOSH is particularly evident as a prebiotic, we do some in vivo tests here. And after the in vivo test, we find that obviously our GOSH is effective in persisting a limited number of microorganisms that we have tested. And from there, we also have the curiosity that, okay, our GOSH is working alone in a good way. How about when it is mixed with some milk or milk proteins? And then where we go is for the in silico modeling of the major milk proteins, that is beta-casein and beta-lactoglobulin, to test that how our glycan protein complex is basically working. And from there, we basically also verify that whether this protein and glycan complex has actually some effect in the probiotic persistence. So with this, I come to my results. So in the first half, we make the galacto-oligosaccharide, characterize that, and in the characterization, this is the NMR graphs you can see. So I have compared it with the commercial GOSH, and we can see that the commercial GOSH only has the beta bonds, whereas our synthetic GOSH, that is our synthesized GOSH, has the diverse alpha and beta bonds. Now, the alpha bonds are also helpful in some way that some of the microorganisms in our gut also can utilize the alpha bonds. So that is also helpful. So the next we do characterization is by Malditov, and we see that there is more similarity in the 2 to 6 monomer units in the commercial and our synthesized GOSH. So coming to the in vitro test, so we tested among around 12 established lab strains, lactobacillus and bifidum strains, and we found that B. bifidum and L. brevis demonstrated the highest growth in our synthesized GOSH. So with this, we also saw the lactic acid and the acetic acid profiles, and we confirmed that the L. brevis A287 produced significant amounts of acetic acid, which is a byproduct and which is very essential for the gut environment in lacto-oligosaccharide, galacto-oligosaccharide, and beta-bonded galacto-oligosaccharides. So the next step was to study it in the mouse models. So in the mouse model, the experimental design was basically first to screen among these two organisms that, sorry, oh, sorry, yeah. So it was to screen among these two organisms that how B. bifidum with GOSH and L. brevis with GOSH is reacting for the survival of our L. brevis and, sorry, for the survival studies. So day 1, day 2, day 3, day 4, all the 4 days, we oral garbaged GOSH with these organisms, and we collected the fecal samples and then tested the survival of these two organisms. From the first study, from this study, we found that L. brevis was the most potent organism, microorganism to survive as compared to B. bifidum. So the next study was, or the persistence next study was only done with the L. brevis, where the day 1, day 2, day 3 was given with L. brevis plus GOSH. However, the day 4, day 5 till day 9, that is the day till the organism persisted, we only oral garbaged with L. brevis. And we did not give the GOSH just to see that how L. brevis is persisting. So the results from that are basically that L. brevis A287 demonstrated higher survival with our synthesized GOSH as compared to the commercial GOSH for the fecal, cecum, and colon samples in all the cases. However, the B. bifidum was not as potent as L. brevis. So for the persistence study, we considered these groups. So these are the sham, that is only the water control, the mono control saccharides, that is the monosaccharides, which had the exactly the same concentration of sugars as we had in our synthesized GOSH. So in this, you can see that the persistence of the red and the blue are with the synthetic GOSH and the commercial GOSH, and they are very prevalent. It is very prevalent that our synthesized GOSH is very much in pace with the commercial GOSH. So the next thing was to see that, okay, our GOSH is working in alone, very good. How about the milk proteins? So just in silico modeling here, and we find that, and the in silico modeling was basically done with two types of milk, goat milk and cow's milk. So goat milk, the broad categories were goat milk, beta casein, goat milk, beta lactoglobulin, and then cow's milk, beta casein, and cow's milk, beta lactoglobulin. So the basic finding from here was that the binding affinity time of bovine beta casein was higher as compared to the goat beta casein. So that obviously showed that there is some pattern difference between the beta casein of goat milk and cow's milk, and also the beta casein and the beta lactoglobulin group. So the in vivo experiments again were done with the mouse trials, where we see that the goat milk, you can see here the goat milk and the cow's milk. The green one is the cow's milk, and the blue one is the goat milk, and the red one is our synthesized GOSH. So as if we compare the two milk categories, cow's milk had a very less persistence to L. brevis. However, the goat milk has a good persistence, and as these were the milks that were only given alone without the S-GOSH, because milk also has some prebiotic oligosaccharides in it. So the next thing was to test that if we add our synthetic GOSH to it, how does that work out? So here we see that when our milk is added with when the milks are added with our synthesized GOSH, the performance of both the milks is substantially increased. However, the goat milk still stays at its best as compared to the cow's milk. So this was the in vitro study to see that how L. brevis and how L. brevis is performing with the individual beta-lactoglobulin and beta-casein, because we isolated these proteins individually from the goat milk and the cow's milk and combined it in a complex to see that how these are reacting. And we see that obviously the results you can see here, the goat milk obviously had a higher persistence even in the in vitro studies as compared to the cow's milk. The GOSH has all the good results for in both the studies. So the final experiment, what we did was to see that how the gauche release because the gauche and the protein are forming a glycan protein complex. So we saw that what is the gauche release time from both these proteins from cow's milk and the goat's milk. And we can see here the gray ones are the casein and the gauche complex, whereas the beta lactoglobulin are the two blue ones. And we can see that it is very evident that the gauche release from the casein protein is very low as compared to the gauche release from the beta lactoglobulin. So this definitely says us that there is definitely a different pattern of the glycan being released from the protein when it is given in a glycan protein complex. So with this, the major conclusions what we had was that utilizing dairy waste, that is from whey permeate to synthesize prebiotic galactooligosaccharides chemically can sustainably decrease the processing costs. However, downstream processes needs optimization for utilization of the chemicals. Our synthesized gauche is an ideal prebiotic for L. brevis out of the 12 probiotic strains that we have evaluated, but it requires testing for other probiotic strains as well. And depending on the type of protein used, the synthetic gauche in combination with milk protein can enhance the quality of milk formulations. And finally, the slow release pattern of the synthetic gauche may be utilized for the carrier of protein molecules in future. So with this, I thank my funding agency, Dairy Innovation Hub, my department, Biological Systems Engineering, our recent collaborator, CDR, my PI, Professor Yuxing Chen, and all the members of the PAN Lab, and my co-PI, Professor Zanwen Pizikaren, and all the members of the VPL. Thank you very much.
I'm pleased to announce our next speaker, which is Maggie Becher. She's a graduate student here at UW-Madison, pursuing her PhD in food science, mentored by the Center for Dairy Research Scientist. Give this a try, Ronnie Govindasamy-Lucy. Did I do okay? Her research involves looking at functional properties of string cheese, like stringiness, as well as flavor changes during storage. And she's also researching how to extend the characteristic squeak of fresh cheese curds. Very interesting.
Okay, thank you for the introduction. Today I'm going to be talking more about squeakiness of fresh cheese curds and some of the research that we've done to try to extend squeakiness shelf life. So how many of you have ever tried fresh squeaky cheese curds? Okay, a really good portion of you, but for those who don't know what cheese curds are, they are typically cheddar and they're typically eaten as a snack. They're pretty basic in terms of flavor, but they're really known for this springy, rubbery texture, which helps to give it this squeakiness. And the squeakier they are, the fresher they are. So a little bit about how cheese curds are typically made. Since they're cheddar, they're typically cultured. So cultures are added to acidify the milk, and then coagulant is added to coagulate the milk, and then the curd is drained from the whey, or the whey is drained from the curds, and then they're allowed to sort of mat together, and then these slabs are milled and then salted, and that gives you your cheese curd. So squeakiness is known as the sharp, high-pitched sound that's produced when the curd rubs against the teeth during during chewing. And the squeak is quickly lost within a couple of days. So this makes it really a local product to the areas where it's typically manufactured. So the main goal for this project is to try to extend the squeakiness shelf life so that everyone can experience fresh cheese curds, and we can distribute it to farther areas around just these local regions. So a little bit about what happens in cheese during storage. So you have proteolysis, which is enzymatic breakdown of proteins. And this can be due to residual enzymes within the cheese matrix. This weakens the protein structure. And also, insoluble calcium is very important to cross-link the proteins, which gives it a nice, strong protein network. And so excessive acidification can solubilize insoluble calcium, which we don't want in this case. So kind of a hypothesis about squeakiness is that this very interconnected and elastic protein matrix is very important because that's what kind of causes this energy buildup as your teeth are sort of passing through the curd. And then once that yield happens, that energy is released as the squeaky sound. So therefore, things like proteolysis and solubilization of insoluble calcium are very important for the cheese curd squeakiness because that's what allows the protein matrix to be very elastic. Kind of an analogy about this, you could think about a balloon. If you've ever seen somebody kind of twisting together a balloon animal, it's kind of squeaky. So if you had a partially deflated balloon, you wouldn't get as much squeakiness. And similarly with the cheese curd matrix, you want a very intact protein structure to kind of help create the squeaky sound. With other types of food sounds like crunchiness or crispiness, you can easily just fracture your product and this creates the sound that you're looking to analyze. However, squeakiness is much more complicated. It's not a brittle product, so you can't just compress it and you get a squeaky sound. There's a lot of things happening in the mouth that contribute to the production of the squeakiness sound. So therefore, our objective is to develop methods to be able to quantify squeakiness. And so we decided to use both acoustic measurements and use descriptive sensory evaluation. And we also wanted to develop methods for looking into the cheese curd texture, which I won't be talking about as much for this presentation. And then we wanted to correlate the acoustic properties with the texture throughout storage. And finally, determine the impact of various levels of proteolysis on the squeakiness and cheese curd texture during storage. So starting out with these manual acoustic recordings, we've been collaborating with an audiology lab at UW-Madison because they have these soundproof booths and sensitive microphones and just proper equipment for us to create these recordings. So in this picture, you can see that we have a microphone placed inside the ear canal. And we tried a few different microphone positions, but this produced the best kind of audio signal that we were getting compared to like a position right outside the mouth or something like that. So next was to gather a lot of squeaky data and a lot of non-squeaky data because we don't really know what these differences are in the acoustic properties of squeaky versus non-squeaky cheese. And so we wanted to gather a bunch of both types of data so that we could eventually classify or train a machine learning model to classify the recordings that we have as either squeaky or not squeaky. So we recruited 7 participants to come in, and they came into the sound booth, they sat down, and we put the microphone in their ear. And then they were looking at this monitor, which I'll show you in the next screen. And then I was sitting outside the booth, kind of directing them. So each participant went through 50 cheese curds. 25 were squeaky, 25 were not squeaky, and they chewed 15 times per curd. And this might sound like a lot of fun, but after 50 curds, it gets pretty tiring. And so calculating all this out, we had over 5,000 possible individual chews to use towards this machine model. Like I mentioned, this was the monitor that the participants were looking at. We used a program called Audacity to record the audio signal. And we also had a metronome set up so that they could like pace their chews. So it was set to 120 beats per minute. So each chew cycle was about 2 seconds long. The reason we did this was so that it would be easier to split up these recordings into individual chews. So we ran each recording through a MATLAB code that detected a local maximum and then split it. If you milliseconds before and after this peak to extract the individual chews. So in total, we had 5,166 files to label. And to label all of these files, the audiology professor I was working with put together the software to help speed things up a little. And so here we're looking at a spectrogram up in the left here, which shows frequency versus time. And the intensity of the color is sort of like the power of the frequency at that time. And in a lot of the squeaky recordings, we noticed a brighter zone from about 1,000 to 2,000 hertz when the squeak occurred in the squeaky files, which I'll come back to in a couple slides. And then if you are interested in hearing what these sound like, so this, I don't know, we'll see how the audio is, but this would be a not squeaky sound. You can kind of hear, it's kind of quiet. And this would be squeaky. So I'll play it again just in case you missed it. That one was not squeaky. And that one was squeaky. So after we had all of these files labeled, we used a long short-term memory neural network to classify a portion of these. So we trained this network on a portion of the files, and then we tested it on a separate portion. And we had a classification accuracy of 86% for the not squeaky files and 94% for the squeaky files. So now that we have this method in place, we wanted to actually use it in a cheese curd trial. So we decided to study the impact of proteolysis on extending the squeakiness shelf life of fresh cheese curds. So for this project, we took inspiration from a cheese called Hustolipa or Husto. It's also known as Finnish squeaky cheese or bread cheese, and this is because it's baked after it's manufactured. So that produces this kind of golden crust that you see on the outside. And the reason why we're interested in Hustolipa is because it's able to retain its squeak for months versus cheese curds, which only last about a week. And this retention of squeak in Hustolipa is likely because there's no acidification during the manufacturing process. And this could help to encourage a higher insoluble calcium content in the cheese. And it also probably has lower proteolysis due to the high heat temperature that it undergoes in the baking step, which can denature the enzymes in the cheese, which contribute to proteolysis. So taking inspiration from Hustolipa, applying it to cheese curds, we decided to make Husto curds. So some treatments here. We did not use any acidification, but we used 2 different types of rennets to look at different proteolytic activities there. And then we either did not bake these curds, baked them to an internal temperature of 60 degrees or 88 degrees Celsius. These curds were then gas flushed, and then the cheese pH was about 6.25 to 6.35, which is very high for cheese. And the cheese was sampled at 2 days, 1 week and 2 weeks of storage, and 1 and 2 months of storage. Here are some pictures. So there wasn't a lot of difference between the rennet types. However, we can definitely start to see a little bit of browning happening in the low bake, but a lot more happening in the highest bake temperature. So for proteolysis in these cheese curds, we expected to see more proteolysis in the no-bake samples because there wasn't that heat step to denature enzymes. And then we saw a reduction in proteolysis due to the low bake temperature, and the highest bake temperature had very low levels of proteolysis. So we also measured the texture profile analysis, and we used the peak force of the first compression as the hardness. So the hardness score, or the hardness values for these cheese curds, we saw that the highest bake temperature had the highest level of hardness. This was expected because it also lost a little bit more moisture during the high bake. And the the firmness really didn't change during storage, but for the no-bake and low-bake samples, we did see a slight reduction in the hardness during storage. So going back to the audio data, I mentioned that we saw that zone of more intensity in certain frequency zones. So we decided to use the average spectral power from 800 to 3000 hertz to sort of quantify squeakiness in these cheese curds that we have been analyzing. And so the second method of analyzing squeakiness, we actually used our trained sensory panelists to detect the squeak intensity. So this was looking at the volume and the prevalence of the squeaks on the first 15 chews, and then they ranked it from 0 to 6, which is from none to pronounced. Looking at these two measures of squeakiness side by side, we see a very similar trend, and we see that overall, the squeakiness did decrease a little throughout storage in most of the samples. However, in the highest bake temperature, there was the greatest level of squeakiness after 2 months of storage. And there was a slight reduction in the low bake samples, there was slightly greater squeakiness compared to the no bake samples. And this is just a correlation between these two methods of squeakiness quantification. Is a pretty high correlation there. We found a moderate correlation between these measures of squeakiness and the TPA hardness. And we saw a pretty strong negative correlation between squeakiness and proteolysis, which supports our hypothesis. So in summary, the highest baking temperature was able to both retain firmness and squeakiness during storage. And the no-bake and low-bake samples softened during storage. However, the low-bake seemed to slightly retain a little bit more squeakiness after 2 months of storage. And we were able to successfully use spectral power as an acoustic squeakiness metric. And we had moderate correlation between texture and squeakiness, and proteolysis was negatively correlated with squeakiness. With that, I'd like to thank everybody involved in this project and the Dairy Innovation Hub for having me here today and funding part of this project, and DMI for additional funding.
So for the squeakiness of the curds after 60 days that became how would that compare to the squeak of like a so is it similar, or would you see still higher levels of squeak in fresh?
They did slightly decrease in squeakiness over time, but so it's not exactly as they were when they were fresh, but there was still some squeakiness left. Yes. Yep. It would be sort of more challenging since cheddar goes through the acidification process, so you're losing some of that insoluble calcium that helps these curds to stay intact while they undergo these high bake temperatures. So with the loss of insoluble calcium, they might actually melt during the baking process, which wouldn't be ideal for this product.
All right, so our final speaker this morning will be Karen Antunes. She's a graduate student in my lab. Her background is a bachelor's degree in nutrition from the State University of Rio de Janeiro in Brazil. She's also a registered dietitian and worked as a nutrition counselor before graduate school. What else? She's the hardest working student I've ever interacted with, I can tell you that. She's in the lab all the time. She's going to be talking about something we haven't heard much about today yet in the talks, but extracellular vesicles, and one creative way that she came up with for treating surgically nutrition-fed animals, which has applications to people. So please help me in welcoming her.
Thank you so much for your kind introduction, Joe. I'm really glad to present our project today, the use of bovine milk, extracellular vesicles in pediatric total parenteral nutrition. So first, you might be wondering, what is surgical nutrition? So we have two types of surgical nutrition. You have parenteral nutrition and enteral nutrition. So parenteral nutrition from the Greek words, para means beside, enteral, intestine. So it's nutrition that is administered directly to the bloodstream when oral or enteral nutrition is not possible due to an incapacity of the GI tract to receive nutrients. And why we are focusing our study in the pediatric population? Because when neonates are born premature with low birth weight, their gut are immature, and they are not ready to receive nutrients through the GI tract. So they have to rely in this first phase into parenteral nutrition to support that growth. And it's really striking and important that it's 34,000 of patients yearly in the United States rely on parenteral nutrition when they have serious health conditions and the neonates when they are premature. So as you can see, parenteral nutrition is a life-saving and is a very important nutritional strategy. It prevents malnutrition, supports the metabolism, and supports the neonatal growth. However, this nutritional strategy comes with serious consequences, and those consequences might be permanent throughout the patient's life. And this type of nutrition might lead to hepatic complications. Well-known in the literature, a condition is PNOW, parenteral nutrition associated liver disease. In the neonates, even though supports growth, not support optimum organ development in the neonates. And for the focus of our project, it alters pro-inflammatory immune response. And also due to the surgical side of it, it has an infectious risk. So we came with this, we have this problem in a life-saving condition. So amazingly, my PI, Dr. Joe Pierre, came create this novel and unique parenteral nutrition model in baby mice. So here you can see is a 17 days old mouse with jugular cannulated, and this catheter goes under the skin and is connected to a button, and this button connects the mouse into a pump that will pump the parenteral nutrition formula continuously. And we had to find a solution to apply to this model to prevent or reduce the inflammatory consequence of parenteral nutrition. So we decided to use a bovine milk bioactive compound. And why bovine milk? So as we all know, humans domesticated cows and consumed their milk starting 10,000 years ago, and milk is a really rich source of nutrition. It's a complete protein, has a stable fat and mineral source. Most of us humans are adapt to consume lactase due to the lactose due to lactase selection, and it is a consistent prebiotic source. And it also provides immunological advantage. So the human FCAR and bovine FZGR2 are a common gene, and they interact and facilitate immune response. And We also cannot forget the main purpose of milk. So milk comes to our gut when we are born sterile to establish and help our gut microbiome to be formed. So beyond all those bioactive compounds and nutrients present in milk, we decided to use the microRNA present in the extracellular vesicles. Now you might be wondering, what are extracellular vesicles? So us human, bacteria, and the cows as well, our cells produce extracellular vesicles. And those tiny vesicles, they are responsible for cell-to-cell communication, and they are rich in microRNA. And this microRNA helps to helps in gene expression, and reduces inflammatory response. So the mammalian cells of the cows, they produce those extracellular vesicles, and those vesicles goes to the milk. And milk is a really rich source of EVs, which I'll call, it's easier to call it EVs. So it's 1 to the 10 particles per ml of EVs in the milk. Even the milk that you find in the grocery stores contained EVs. So our first step through this project was to get these EVs out of the milk. And this was a brilliant work by Cameron Latham, and he isolated the EVs out of the milk after a series of centrifugation, pH treatment, and filtration. We were able to isolate the EVs, but we need to make sure if what we got were the EVs that we are looking for. So first, the EVs, they're in the range of size between 50 to 200 nanometers. And we sent our EVs to NTA to confirm the size, and it's 106, I know it's pretty small. So we confirmed that we had the right size. And Cameron also performed a Western blot with protein targets that are present in the EV's membrane. And we are able to identify those. And also, we were able to see that we had no contamination. Our negative connexin control came clean. And we also send our EVs to TM to analyze morphology. And you can see here the lipid bilayer, how they look. And the most interesting part of this analysis, we sent to RNA sequencing. And 48% of the microRNA in the EVs are from the family LAT7. And what is interesting about LAT7 is that they are important immune response modulators.